Hello everyone, welcome back to the video presentations. Today we will discuss the regulation of gene expression in prokaryotes, and we will be focusing on basically on the mechanism of lac-operon and trip-operon. So before moving into the mechanisms of lac-operon and trip-operon, we will see some general concepts of gene expression regulation. So we know that gene expression is the basis of expression of a character, or a character is nothing but what genes are present in a system, or what genes are present in a cell, and how they are expressed, or which genes are expressed at a particular time. So the basic reactions that regulate or that control the gene expression are, as already we have studied, as the transcription and translation. These are the two events that leads to the reaction, or that leads to the expression of a gene, or that ultimately gives you a character. So we know the basics, like mRNAs are produced after transcription, and these mRNAs will be translated to the proteins, and these proteins will perform a particular function, and this function represents a character. And as already we have seen in the previous videos, that in prokaryotes the translation is usually coupled to the transcription. But in case of a eukaryotic system, you can see a little more elaborated processing mechanisms before it is subjected for translation. So this is the basic idea of gene expression. These proteins will do the biological functions. Then out of the various number of genes, not all genes are expressed all the time, but instead certain genes are expressed at certain times and also at certain cells. That means certain genes, even though we have around 20,000 to 25,000 genes in human chromosome, not all these genes are expressed in all the cells at all the time, but there will be expression of certain genes in certain types of cells, and moreover there will be expression of certain genes at certain times only. For example, there will be certain genes which are expressed only in the developing cells, right? And there will be certain other genes which are expressed in only in an immune cell. There will be certain other genes which are expressed in a particular organs cells, like an enzyme which is required by the liver cells will be synthesized only by the liver cells, right, not the other cells, which doesn't mean that that particular gene is not present in another organs or another cell in the body. The gene will be there, but the expression is under regulation. You can see a lot of examples, like we know that the insulin is produced by the pancreatic cells, right, and the gene for the insulin production is, of course, will be present in the beta cells of the pancreas, right, which means that the gene for the insulin production is there in that particular cell or in the chromosome, which also says, or which also we can understand that these genes, the gene for the insulin, will be present in other cells of the body also. That means in the intestinal cells, in the epithelial cells, in the kidney cells, in other liver cells, in all cells you can see the copy of the insulin gene, but these genes are not expressed in cells other than the pancreatic system. So these genes other than the pancreatic cells are under regulation. They are not expressed at all. Okay, so in this way you can regulate, or you can control the characters. And gene expression could be of basically of two types: constitutive gene expression and regulated gene expression. The constitutive genes are nothing but those genes which are permanently expressed. There will be certain genes which are required by the cell most of the times. In that situation, these genes are not under regulation. So such genes are called housekeeping genes, and Such enzymes or proteins are called housekeeping proteins or housekeeping enzymes, right? You can find examples like a glycolytic pathway. So the enzymes required for glycolytic pathway are expressed all the time. That means at any point of time in a bacterial system, when you analyze, you can see all the enzymes required for glycolysis. So these genes are expressed most of the time, right? So these enzymes are called housekeeping enzymes. At the same time, there will be certain genes which are under regulation. They are called as regulated gene expression. So these gene products are required based on the molecular signals received by the cell, right? So the concentration may either rise or fall based on the nature of the signal. At certain times you need a higher concentration of a protein, and certain other times you need a lower concentration of that protein. So this control, or this concentration, this concentration will be maintained by certain molecular signals. All right. So if the concentration increases, then such genes are referred to as inducible genes, and if the concentration decreases, then they are represented as the repressible genes. So an inducible gene will be expressed in response to a signal, and that particular mechanism is usually we call induction. So induction is the process by which a gene is induced to express, right? So the genes will start to express and produce the proteins, and there will be functions will be performed, right? At the same time, a repression is the process by which an already expressing gene will be repressed, right? That means a repressible gene will stop expression in response to a signal, and that process is called repression. In otherwise, in short forms, you can see the image also. Induction is the process in which the gene which is not active or which is not in the expressed state are subjected to expression by a process called induction. At the same time, repression is for a gene which is already in the expressed status, and these expressing genes are given certain negative signals so that the expression will be limited or inhibited. So this is what induction and repression. Most of the inducible enzymes will be working in catabolic pathways, that means in the breakdown pathways. So their synthesis is induced by certain chemical signals, and these genes has to be activated only when the corresponding substrate is available to the cell. You can see the classical example as the lactoperon, in which the breakdown of lactose will be activated only when a lactose is supplied in the medium. So lactose is the inducer here, and this will induce the operon called lactoperon, and that leads to the production of enzymes which are required for the breakdown of lactose. So that is all about inducible enzymes or inducible pathways. Repressible enzymes are usually will be functioning in anabolic pathways, that means in the synthetic pathways, right? So when the product of the pathway is available in excess quantities, then there is no need for further synthesis. So in such situations, the system has to be shut off. And here we have the representative example as the tryptophan. Here the tryptophan has to be synthesized by the tryptophan. So when tryptophan is abundantly available, then there is no need for the synthesis of tryptophan. So in that situation, the tryptophan will act as a repressor, and they will repress the tryptophan. So that's about repressible pathways and repressible enzymes. So in the coming sessions, we'll discuss in detail about lactoperon and tryptophan, and I hope you understood these basic concepts. Thank you for listening.